glycolysis, correct na? So tell me all of you, in case of a glycolysis, how many number of, slowly think and answer, gross ATP can be produced? Gross. What is the difference between the gross and net? Net is visible. How many you can see is the net? Gross is what? Gross means, gross represents what? Total. Okay, total is called as the gross. Okay, when I consider the gross, what I will consider? I will consider 2 molecules of NADH and 2 molecules of ATP. Okay, but when I consider the net, I consider only 2 molecules of the ATP. Okay, that's the fundamental difference. Now, where the process of the glycolysis taken place? Why only in the site? What is the difference between cytosol and plasm?
Cytosol, if you remove organelles.
Correct. So cytoplasm is a liquid or a matrix or a gel which keeps on moving and holding the organelles. If I remove all, or if I separate the organelles from the fluid, left out is cytosol. So it occurs generally in the cytosol, not in the cytoplasm. Why glycolysis only taken place in the cytosol, not in any other area? Because all the enzymes are present. And the most important question, all of you answer, what are the 3 enzymes which are said to be the regulatory enzymes of the glycolysis? Not even 48 hours. How come you forgot? Sanjana, what are the 3 enzymes? Number 1 is hexokinase. Number 2? Very good. Phosphofructokinase. Number 3? Number 3, most important one, pyruvate kinase. Correct na? Pyruvate kinase. Tell me why we have used the word regulatory enzymes? Because they are irreversible. They are going to perform the reactions which are completely irreversible. Now what the question which is asked in the exam, why, among these 3, which is the rate limiting enzyme? What is the reason why did I call rate limiting enzyme? What is the function of this enzyme specifically? Conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. And tell me why did I use the word rate limiting? Because if this enzyme is absent, there is no other enzyme that can convert fructose 6-phosphate to fructose 1,6-bisphosphate. Moreover, you cannot produce the fructose 1,6-bisphosphate in the absence of this enzyme. Now, why we have not used the word hexokinase and pyruvate kinase are not rate limiting? What is the function of hexokinase? Tell me, how can I produce the glucose 6-phosphate if hexokinase is absent? We already studied so many times. Whenever we break down the glycogen break down into glucose 6-phosphate. Later on it can give rise to. So what my conclusion here, even though hexokinase enzyme is absent, I can produce 6-phosphate. Hence, it is an important enzyme but not rate limiting. And what about the pyruvate kinase? What is the function of it? Conversion of phosphoenol pyruvate to pyruvate. So tell me, how can I produce the pyruvates? Consider this enzyme is absent. I can produce the pyruvate from many amino acids. Especially alanine is the name of the amino acid that can give rise to. Hence, even though this enzyme is absent, I can produce. But if PFK is absent, I cannot produce. So the conclusion here, there are 3 regulatory enzymes which are irreversible in nature, but one is rate limiting enzyme. Wonderful. Okay, that is what we have discussed. Now finally friends, please summarize the equation of the glycolysis. What happened friends? We break down 1 glucose into how many pyruvates?
2 pyruvates.
Glucose is how many carbon?
6 carbon.
6 carbon. Giving rise to how many pyruvates?
2 pyruvates.
Each pyruvate is how many carbon?
3 carbon.
3 carbon. Okay. And what else we get? 2 pyruvates. Very good. 2 ATPs. Very good. 2 NADH and 2 water molecules. I told you not to remember 2, 2, 2, 2. Okay. 4 2's will come. Okay. That is what we have studied in the last class. Now friends, Tell me, what is the ultimate goal of the cellular respiration process? Very good. Ultimate goal of the cellular respiration is oxidation of, oxidation means oxidation, breaking. Every biomolecule or every food particle that we consume, it will break down and going to give rise to the energy. Similarly, glucose undergo the mechanism of oxidation and giving rise to 36 to 38 molecules of ATP. But what we people understand actually, we understand that glucose cannot produce 36 to 38 molecules of ATP in one step. There are multiple steps are there. What are those? Glycolysis, Krebs cycle, ETC and oxidative phosphorylation. All 4 steps together it is going to give rise to 36 to 38. But so far what we discussed in the previous class is just glycolysis. Hardly we produced 2 ATP. Okay. Now never ever get confused with the statement. Always try to remember this statement. What are NADH and FADH to Kusma? What do we call them? Tell me man, Kunal, what do we call these two? Very good. See, they know they are not sugars. Okay. Whenever we use the word ATP, what we say? Energy equivalent or energy currency. Similarly, we can say reducing equivalent. Of course, they are going to produce the ATP but not directly. They are the indirect form. Generally, we called as reducing equivalent. Most widely, we called them as electron. See, why do we use the word electron transport chain? So, someone is carrying the electrons into the inner membrane of mitochondria. Who is carrying the electrons? NADH and FADH2. Anyway, now focus from this particular slide, very elaborative, and I want you to solve all the previous year questions correctly. So, we completed the number 1 step, that is glycolysis. All of you focus on the picture so clearly. Now, finally, what we produced? Pyruvate. Pyruvate is how many carbon?
3 carbon.
How many pyruvates we got?
2 pyruvates.
Okay. Now, this pyruvate having variety of fates, and this fate of the pyruvate exclusively decided by the presence and absence of oxygen in a different organisms. Correct na? Now, pyruvate is how many carbon? 3 carbon. We will discuss very elaboratively. Focus. This is 100% conceptual, very small amount of information to be remembered. Pyruvate in the presence of oxygen going to give rise to the acetyl CoA. Acetyl CoA is how many carbon? This is CoA, ma. CoA is not a carbon. Okay. So 1, 2. Correct na? So now consider 1 pyruvate given rise to, you want turn off? Turn off, men. Where is the AC remote? Turn off. That one. Tell me, 1 pyruvate is going to give rise to what? 1 acetyl CoA. How many carbons are left out? Ma, 1 pyruvate is 3 carbon. 1 acetyl CoA is how many carbon? 2 carbon. Then how many carbons are left out? Releasing in the form of CO2. Carbon dioxide. So, but in the glycolysis, how many pyruvates we produced? 2 pyruvates are going to give rise to how many acetyl CoA? 2 acetyl CoA, and there is a release of 2 CO2. Correct na? And the most important, we'll discuss in the upcoming slides elaboratively, conversion of the pyruvate into acetyl CoA. It is a reduction reaction, which means there is a production of reducing equivalent. Obviously, what is getting produced here, friends? NADH2 is getting produced. Now the interesting point, you already know that 1 NADH2 give rise to
3 ATP.
So how many pyruvates we have? 2. So undoubtedly we get 2 NADH2. So this is only in the presence of oxygen. Now let's consider in the absence of oxygen. Again, the fate of the pyruvate can be determined in the absence of oxygen in a different ways in different organisms. Let's consider pyruvate in the absence of oxygen, the organisms such as yeast. It is converted into what? Ethanol or ethyl alcohol. So how many carbons it is?
2 carbon.
So again, when pyruvate converting into ethanol, how many carbon dioxide released for 1 pyruvate? 1 carbon dioxide. For 2 pyruvates, 2 carbon dioxide. Correct, na? Now friends, once again understand, this is what reaction? That's why we produced NADH. But when I use the word fermentation, it is a oxidation, where NADH2 oxidizes means breaking down to NAD plus. You are getting the point. Now the same pyruvate in case of a muscles. See, whenever we are doing a vigorous exercise, continuous cycling, running, weightlifting, all of a sudden muscle starts getting cramp. So what's happening over there? The pyruvic acid, or in simple words, pyruvic acid is replaced by lactic acid because of the absence of oxygen. So when I take a deep breath or when I take a rest for some time, automatically we get enough of oxygen. Then what happened to that lactic acid? Lactic acid enter into the liver, and in the liver again it is converted to pyruvic acid, and again it enter into the glycolysis. Now we will see that. But the conversion of pyruvate to the lactic acid is only happening where? In the skeletal muscle. Pyruvate is how many carbon?
3 carbon.
Lactic acid or lactate? 3. 1, 2, 3 carbon. Hence, is there any release of the carbon dioxide? No. This is the exact question in the NEET. In which of the following reaction there won't be a release of carbon dioxide? Only whenever the pyruvate converted into lactic acid, because pyruvate 3 carbon, this is also 3 carbon, and this is also oxidation reaction where NADH2 break down to NAD plus. So there is only one reduction, and that reduction happen only in the presence of oxygen. No point of confusing over there. So all of you just try to understand. Question won't go this beyond this PPT. It is only more than enough. Read it correctly. 2 molecules of 3 carbon pyruvic acid are generated at the end of incomplete oxidation of 6 carbon glucoses in glycolysis. Correct, na? Each pyruvate is how many carbon?
3 carbon.
3 carbon. So the fate of pyruvic acid is exclusively depending on the need of the cell. Correct, na? Majorly decided by absence and presence. Only in the presence pyruvic acid converted into what? Acetyl CoA. Only acetyl CoA can enter into the Krebs cycle, where there is a release of carbon dioxide followed by the water. What happens in the absence of the oxygen? In case of yeast, converted into lactic acid, sorry, in case of humans, muscles, it is converted into lactic acid, and in the yeast it is converted into the ethyl alcohol. Okay, now look into the point. I'll explain everything simply. So first, pyruvate is how many carbon?
3 carbon.
3 carbon, correct, na? Now 3 carbons, what is the reaction, friends? It is oxidation or reduction? Oxidation. It is not a reduction. Sorry, yeah, correct. Now pyruvate is oxidation to what? Ethanol. How many carbons are left out? 1 carbon. Released in the form of carbon dioxide. So, but friends, pyruvate cannot converted into ethanol directly. You are getting? So, if you observe the picture, we will see it elaboratively. See here. First, there is a mechanism of decarboxylation, then there is a reduction, then only we get the ethanol. Okay, first listen. If you understand and if you read it, it becomes easy. So, see here, there are 2 enzymes are involved in converting the pyruvate to the ethanol. Number 1 is, what is the meaning of a decarboxylase? Which removes the carbon. Then there is another one is called alcohol dehydrogenase, which is involved in the production of the alcohol. Okay. The process of alcohol fermentation broadly divided into glycolysis, decarboxylation and reduction. Anyway, in the glycolysis, what do we got, friends? 2 molecules of pyruvate. Now what happened to that pyruvate now? Undergoes? So which means it is losing 1 carbon. Since we get 2 pyruvates in the glycolysis, 2 pyruvates are going to release into 2 carbon dioxide. Since I am removing the carbon, the enzyme involved is? So where the difficult question comes from? They will ask the question, which of the following cofactors required for the action of pyruvate decarboxylase. Or they'll give a matching question, they'll give 5 to 6 cofactors in one column and they'll give the enzymes in the other column. So there are two major cofactors. One is called TPP, thiamine pyrophosphate, and zinc. All of you answer. In the glycolysis we have 10 enzymes. Which cofactor is most widely used by most of the enzyme? Which is the magnesium. Correct na? Here pyruvate, in order to convert into the ethanol or ethyl alcohol, the enzyme using TPP and zinc as their cofactors. So finally what happened, friends? Which is the reduction. So first pyruvate giving rise to the acetaldehyde. Correct na? By a mechanism called as decarboxylation. First pyruvate giving rise to acetaldehyde, then finally acetaldehyde undergo the process of reduction and giving rise to what? Ethanol. In a simple words, remember. So first pyruvate converted into acetaldehyde by an enzyme. Very good. Then acetaldehyde is undergoing the reduction and giving rise to ethanol by an enzyme.
Dehydrogenase.
Very good. Alcohol dehydrogenase. Very good. Cofactor will be the zinc. Very, very good. What man? Yeah, thiamine pyrophosphate. Okay. Okay. Now everyone just try to understand the next one. What do we call as? Lactic acid fermentation. Where it will occur generally? Lactic acid accumulation majorly occur where? In the skeletal muscles. Correct na? And that too, this particular pyruvic acid cannot be converted into the lactic acid all the time. It only accumulated during the strenuous exercise. Only during that time it will accumulate. Now again glucose giving rise to the pyruvate converted into the lactate. Correct na? Again once we take a fresh oxygen, again lactic acid come back to the pyruvic acid. Don't confuse over there. And this process also happens in the liver. Correct na? Whenever there is a accumulation of the lactate, muscles become tired and become completely painful. When we take a fresh oxygen, automatically oxygen concentration increases, lactic acid is immediately replaced by pyruvate. Correct na? So very simple. Again look into the picture. First glucose given rise to pyruvate. How many pyruvates we got? 2 pyruvates. Now once again don't get confused here. We'll discuss that. Now this particular pyruvic acid converted into what? Lactic acid. There is no change in the carbon number because pyruvate is 3 carbon, lactic acid is also a 3 carbon. Correct na? Now I will tell you in the upcoming slide. Pyruvate converted into acetyl CoA by an enzyme called as pyruvate dehydrogenase enzyme. Okay, similarly pyruvate converted into lactic acid by lactate dehydrogenase enzyme. So both enzymes are the dehydrogenases enzymes only. No point of getting confused. Okay, so if you read it, you'll understand it very clearly. Okay, now majorly compared to the anaerobic respiration, we should always focus on the aerobic, because if we continue the anaerobic respiration, we get very less amount of energy, but our goal is to produce bulk amount of the ATP. Then obviously pyruvate mostly follows the aerobic reaction. So once again focus, 2 pyruvic acid break down into 2 acetyl CoA. Correct na? See, that is pyruvic acid. When 2 pyruvic acids are combined with each other, we get the glucose molecule. Okay, this reaction is wrong. Leave it. I'll explain it in a simple way. Not required that one. Look into this. Pyruvate converted into what? Acetyl-CoA. Okay. One pyruvate is 3 carbon. So this pyruvate giving rise to the acetyl-CoA by an enzyme called as pyruvate dehydrogenase complex. Now remember, this complex is made up of 3 subunits. One is called E1, another one is called E2, another one is called as the E3. Right. So these 3 are said to be 3 subenzymatic complexes. In those 3 enzymes, the number 1 is pyruvate dehydrogenase E1 or enzyme 1. Second one is called dihydrolipoyl transacetylase, number 2, and number 3 most important one, dihydrolipoyl transacetylase. So I will tell you in a simple words, you people remember like this. Number 1 enzyme, dehydrogenase E1. Then number 2, you remember, dihydrolipoyl transacetylase, which is E2. Okay. Then dihydrolipoyl dehydrogenase, this third enzyme, which is called as E3. Okay. It is not a transacetylase, only there is one transacetylase and 2 dehydrogenases enzymes. That is how pyruvate converted into what? Acetyl-CoA. Correct na? Now again friends, for the exam point of view, when pyruvic acid is going to give rise to the acetyl-CoA, the enzyme involved is pyruvate dehydrogenase, but there are 3 enzymes are there. So every enzyme uses different type of a cofactors. Again remember, we require magnesium, lipoic acid and TPP. Okay, undoubtedly they will raise the question in the exams. Required magnesium, required lipoic acid and required TPP as their cofactors. Now, finally what happened friends? Finally this particular pyruvate converted into the acetyl-CoA. Acetyl-CoA is how many carbon? 2 carbon. Now there is a simple question for the exam. Now my question is super clear. Where pyruvate converted into the acetyl-CoA? Pyruvate production occur in the okay, or cytosol. 90% of the students will confuse that pyruvate converted into acetyl-CoA within the cytosol and then they enter into the mitochondria, which is the wrong statement. So look into the point. This is not required, but understand. This is the outer membrane, this is the inner membrane of the mitochondria. Everyone remember, right? You read in the cell biology also. Outer membrane of the mitochondria is more permeable compared to the inner membrane. What happened? On the membrane of the mitochondria, we have certain transporters named as pyruvate translocase. What is the function of that pyruvate translocase? Which allow the pyruvate to enter into the matrix. Why it entered only into the matrix? Because what is the enzyme that breaks down the pyruvate into acetyl-CoA? Pyruvate dehydrogenase. The dehydrogenase is present in the matrix. So within the matrix only pyruvate break down into acetyl-CoA. Correct na? Now what happened to that particular acetyl-CoA? That is the one which is entering into where? Krebs cycle. We read now in the first class whether it is a fat, whether it is a protein, whether it is a carbohydrate. Ultimately everyone is going to give rise to what? Acetyl-CoA. That's why we will discuss that. That's why acetyl-CoA is said to be the important intermediate molecule involved in the metabolism of all the biomolecules. Any biomolecule wants to give rise to the abundant amount of the energy, they give via, because pyruvate can't enter into the Krebs cycle. There is only one product enter, that is, so undoubtedly fat giving rise to pyruvate, pyruvate converted to protein breakdown into amino acid undergoes transamination, giving rise to pyruvate, pyruvate converted into carbohydrates such as glucose, ultimately going to give rise to pyruvate followed by the acetyl-CoA. Correct na? So now focus all of you. Glycolysis which occurs in the cytoplasm is a common pathway in both Aerobic and anaerobic respiration. Here in case of a aerobic respiration, when I use the word aerobic, always remember complete oxidation occur. Whenever there is a complete oxidation occur, what do we get?
Pyruvate.
We get the pyruvate. So for aerobic respiration to takes place within the mitochondria, so look into this. The final product of the glycolysis, that is
Pyruvate.
has to transported from the cytoplasm into how it is going to transport? Via translocase, which is called carrier, which is located on the outer membrane of mitochondria, which can easily allow it. So finally where the pyruvate entered? In the mitochondria. So friends, now don't get confused. What happened finally to the pyruvate or pyruvic acid finally converted into what? Acetyl CoA. Correct na? So since in the glycolysis we produced pyruvate. Pyruvate converted into and the acetyl CoA enter into Krebs cycle. Since the acetyl CoA is produced between the Krebs cycle and that is glycolysis, after the glycolysis and before the that's why this acetyl production of acetyl CoA act like a link reaction. Correct na? Now the question for the exam, answer all of you. Conversion of pyruvate to acetyl CoA mediated by which enzyme? Pyruvate. Now the question is conversion of, you have to tell it the statement is true or false. Conversion of the pyruvate to acetyl CoA is strictly a carboxylation process happening in the mitochondrial matrix. Once again listen the statement. Conversion of the pyruvate to acetyl CoA is a carboxylation process strictly happening in the mitochondrial matrix. Why? Which is decarboxylation, because we are removing the carbon. By that pyruvate can be converted into what? Acetyl CoA. Correct na? So how acetyl CoA can be formed? Acetyl group adding to the coenzyme A. That's why we call link. Since this link is formed in the intermediate stages, we generally call it as the link reaction. Acetyl fraction formed after decarboxylation is linked with coenzyme A and there is a formation of acetyl CoA. That's why we can call it as a link reaction, or we can also call it as acetylation of pyruvate. Correct na? Pyruvate undergo the acetylation given rise to the acetyl fraction combined with the CoA. No point of confusing over there. Now first I'll explain the Krebs cycle completely, then we'll read the theoretical information so easily. Now I want everyone to answer every single question correctly. Okay. Now look into this line what we have written here. Glucose converted into pyruvate and converted into acetyl. Fat also same. What is the name of the breaking down of or oxidation of fatty acid called as? We read now in the last class, breaking down of the glucose is called as glycolysis. Correct na? Breaking down of fats called as beta oxidation. Correct na? Beta oxidation is the name of the mechanism by which fats break down into glucose or pyruvate we can consider. Okay. Now all of you focus on the picture so clearly without having a confusion. Just understand the picture as it is. How many acetyl CoA we produced? Two. So while considering, we'll consider for 1, while calculating, we'll consider for 2. So slowly understand the concept correctly. Let's talk about 1. So 1 acetyl CoA is how many carbon? 2 carbon. So the first, so someone has to accept the acetyl CoA. Who accepted the carbon dioxide in C3 pathway? Remember? RUBP. In C4 pathway? Pepcase is an enzyme. Rubisco is also an enzyme. RUBP is an acceptor. In case of CAM pathway also same. PEP only, we will read that. Similarly here, who is accepting the acetyl CoA? Oxaloacetic acid. Oxaloacetic acid is how many carbon? 4 carbon. So 4 carbon containing compound combined with 2 carbon. Acetyl CoA is 2 carbon. So we produced 6 carbon. So the first stable or intermediate product to produce this the 6 carbon, that's why we call it as citric acid cycle. Also called as Krebs cycle, also called as, why do we call TCA, man? Why?
3 times
Why? Why? See, what is the first product? Citrate or citric acid. The citrate or citric acid having 3 carboxyl groups. How many? 3 carboxyl groups. That's the reason we also called as tricarboxylic acid. Tricarboxyl doesn't mean that the cycle is happening twice, because the first stable product contains 3 carboxyl groups. Correct na? Now, everywhere first product is very important. Why we have given the name C3 pathway? Based on the first product only. C4 pathway based on the first product. So any pathway first product or intermediate is very important. Anyway, so citrate is how many carbon?
6 carbon.
So now citrate convert into isocitrate is how many carbon? 6 carbon. No change in the carbon number. Correct na? So citrate convert into its isomer isocitrate, which is ecotinase enzyme. Okay, type of isomerase enzyme only. Now isocitrate is how many carbon? Converted into alpha ketoglutarate is how many carbon? 5 carbon. Which means, friends, what happened, tell me? Very good. There is a loss of 1 carbon. See, I always remember, I told you in the first class, wherever you heard the name of a dehydrogenase, always there is a loss of carbon. Mostly there is a production of either NADH or FADH2. I am giving a hint. Wherever you heard the name dehydrogenase, there is a production of reducing equivalent. Sometimes it may be NADH or sometimes it may be FADH. So all of you focus. So 6 carbon converted into 5 carbon. There is a loss of how many carbon? I am calculating for 1:1 co-aid. So how many carbon are lost?
1 carbon.
And it is a reduction reaction where there is a production of
NADH.
So first reduction reaction it is. You have to remember that. Correct na? And what is the enzyme involved here? Isocitrate dehydrogenase, because we are reducing the isocitrate. That's why isocitrate dehydrogenase, first reduction reaction, loss of 1 carbon dioxide and production of 1 NADH. Alpha ketoglutarate is how many carbon?
5 carbon.
Converted into succinyl CoA is how many carbon? 4 carbon, which means there is a loss of 1 more carbon in the form of carbon dioxide. Again reduction reaction, where NAD plus reduced to and again, since the NADH is produced, the enzyme involved is what got undergone the decarboxylation? Alpha ketoglutarate. So enzyme is alpha ketoglutarate dehydrogenase. So this is the second reduction reaction. Now succinyl coenzyme is 4 carbon converted into succinate is how many carbon?
4 carbon.
No change in the carbon number. Now question is super clear. Here there is a production of
GTP.
How GTP got formed? GDP combined with
Phosphate.
which is called inorganic phosphate. Now question is super clear, from where this PI came from. See, coenzyme A contain a bond named as a thioester bond. Okay, thioester bond is present in the coenzyme A, which is energy storing molecule. Whenever there is a breakdown of the succinyl coenzyme A, this thioester bond will break down, which generates energy, and someone accepts that energy to combine GDP with PI, then there is a formation of
GTP.
We already studied in the first class, 1 GTP is going to convert into 1 ATP. Now why cells won't use GTP? Why they are using ATP directly? Because most of the enzymes or most of the reactions having binding sites for only ATP, not for the GTP. That's why they use it. Must switch on AC sometime. Okay, now succinate is how many carbon now? 4 carbon. Now succinate converted into fumarate is how many carbon?
4 carbon.
No change in the carbon number. Correct na? Now when succinate converted into the fumarate, again it is a reduction. But this time the reducing equivalent is FADH2, not the NADH. Now you can ask me, sir, what is the reason? Because the enzyme, which is succinic dehydrogenase, it is having the binding site for FAD, not for the NAD. Right? Then FAD reduced to FADH2. Anyway, you know that 1 FADH2 give rise to 2 ATP. Fumarate is how many carbon?
4 carbon.
Fumarate converted into malate how many carbon? No change in the in the carbon number, just fumarate convert into malate by the addition of water. Just hydration. Then convert into malate. Malate how many carbon?
4 carbon.
Malate converted to?
Oxaloacetate.
Again 4 carbon, 4 carbon, no change in the carbon number, again reduction. Reduction means the enzyme is
Dehydrogenase.
what we are reducing?
Malate.
Then there is a production of
NADH.
1 NADH. Now tell me, in the entire Krebs cycle, how many reduction reaction occur?
4.
4 reduction reactions occur. Correct now? No point of getting confused. Now 90% of the time questions never appear from the structures, but there is a possibility, and it is very difficult to remember every structure. So the best way of remembering is carbon numbers. In the entire cycle, how many 5 carbon containing compounds are there? Only 1. Easily you can remember. In the entire cycle, how many 6 carbons are there? 2. One is citrate and one is isocitrate. And how difficult the structure it may look like, if you remember CoA, that will be succinyl CoA. So in the structure you can see the CoA. Okay. Then only you have to remember succinate, fumarate, malate, oxaloacetate. All remaining are 4 carbon numbers. Correct na? Easily we can understand. So now I will tell you how exactly to do the calculation. Later we will read about every single enzyme separately and we will try to understand this. Now, don't get confused. Okay, whenever you are doing the calculations, you have to do it in a very different way. Let's consider different type of steps. Let's consider direct ATP, GTP, FADH2, NADH2, total. Simple calculations only will do. So let's consider the first step.
Glycolysis.
What is the total net ATP produced? 2. Correct na? Then is there any direct or indirect GTPs? No. FADH2? No. NADH? 2. So but I told you na, according to the recent calculations, they may consider 1 NADH2 into 2.5 also, 3 also. FADH2 into 1.5 also, 2 also. That you have to calculate based on the answers given. Then 1 NADH2, 3 ATP we will consider. Then total 6 plus 2, 8. 8 means what, friends? Gross. Why I taken gross? Why I did not use a net now? Because I am calculating for 1 glucose. Completely I am talking about complete oxidation of glucose. What is the next step? After glycolysis, Krebs cycle. But before entering into the Krebs cycle, link reaction, which is called as pyruvate decarboxylation. Don't use the word link reaction all the time because it's a common term. Remember the new term, pyruvate decarboxylation. Correct na? Now in the link reaction, is there any production of ATP? No. GTP? No. FADH2? No. NADH2? 2. Correct na? When 1 pyruvate reduced to 1 acetyl CoA, 1 NADH. 2 pyruvates, 2 acetyl CoA. 2 NADH. So 2 into 3? 6. Correct na? Now, how many acetyl CoA entered into the Krebs cycle? Both 2 entered. But we'll consider for 1, we'll calculate for multiply for 2. So I am taking for Krebs cycle for 1 acetyl CoA. Aram se you have to think it. 1 acetyl CoA, 1 GTP we produced. Correct na? 1 GTP represents 180. So I am writing only 1. Correct na? So anyway, GTP only I consider it as the ATP. No direct ATP produced. Correct or not? That is most important point. Compared to the pyruvate dehydrogenase pyruvate decarboxylation and Krebs cycle, direct ATP produced only in the glycolysis. Correct na? Now don't get confused. How many? So no 1 GTP, that only we convert into ATP. How many FADH2 for 1 acetyl CoA? 1. 1 into 2?
2 ATP.
2 ATP. How many NADH? 3. Because there are 3 reduction, total 4, and among those 3 reduction reactions are NADH2. 3 into 3?
9.
So 9 plus 2 plus 1. How much? 12. 12 for 1 acetyl CoA. When I consider for 2 acetyl CoA, 12 into 2, 24. So 24 plus 6 plus 8, 38 molecules of ATP. Again, there is a small difference. When I use the word gross ATP per oxidation of one glucose molecule, per oxidation of one glucose molecule, gross is 38. Again, they may raise the question, what is the net ATP? 36. What happened to that remaining 2? Used in electron transport chain. Okay, we'll discuss that how. Now tell me, now don't get confused. Okay, aram se think and answer. Give me the total number of NADH, FADH2, GTP per one oxidation of 2 glucose molecules in Krebs cycle. Once again, listen. What is the total number of NADH, FADH2 and GTP that are produced in the Krebs cycle per oxidation of 2 glucose molecules? How much?
6 plus 6, 12.
Aram se.
NADP 12.
I asked for how many?
2 glucose.
2 glucose.
4
And I asked for where? I asked for which cycle? Krebs cycle. Never consider the number of acetyl CoAs here. Consider the number of cycles. I asked for 2 oxidation of 2 glucose molecules. Now tell me, 1 glucose molecules means how many pyruvates? 2 pyruvates. 2 pyruvates means 2 acetyl CoA. Then you don't need to consider for 1 acetyl CoA because I am asking for 1 glucose or 2 glucoses. Tell me. For 1 glucose, how many cycles?
2 cycles.
Very good. 2 cycles. 2 glucoses means 4 cycles. That's all. So 1 cycle, 1 glucose, how many NADH? For 1 glucose,
36, sir, for 4.
tell me for 1 enough.
9.
Tell me for 1 glucose, how it is 9?
3. 1 glucose 6.
1 glucose is how much ma? 6.
12.
6 NADH. Here
2.
2 FADH2. GTP? 2. Very good. Correct or not? Now let me ask in a different way. Tell me for 1 acetyl CoA molecule.
1 acetyl. 3
3 NADH, 1 FADH2, 1 ATP. Consider it as the ATP. Now we can easily say 1 glucose, 2 acetyl CoA. Correct or not? So for 2 acetyl CoA, obviously 6 NADH, 2 FADH2 and 2 ATP molecule. Now don't get confused and answer my question. Per 1 glucose molecule, I am asking per 1 glucose. Oxidation of 1 glucose molecule, what is the total number of NADH?
Total number of NADH.
Per oxidation of 1 glucose, I am asking.
8.
Aram se man, aram se. Per oxidation of 1 glucose. When I use the word oxidation of 1 complete glucose molecule, should I consider all the steps or not?
Yes.
Correct na? Then glycolysis, how many we produced? 2. Link reaction.
6.
Link reaction 2. Aram se. Okay. Then Krebs cycle we produce how many? 6. You getting or not? So for 1 glucose we get 10 NADH. Now tell me for 1 glucose how many FADH2? 2. That's all. Only 2. Per 1 glucose how many GTP? Per 1 glucose. 1 glucose is 2. Correct na? So now, very good, all are correct. Okay. So in a very simple words, never ever remember as acetyl CoA molecule, remember for glucose. They won't ask the question for acetyl CoA. Anyway, you can remember acetyl CoA also. So in simple words, 1 glucose, 2 pyruvates, 2 acetyl CoA. Correct na? When I consider for 1 acetyl CoA, how many NADH? 3 NADH, 1 DH2 and 1 GT. This is for 1 acetyl CoA, not for 1 glucose. When I use the word 1 glucose, 6 NADH, 2 FADH2 and 2 GT. So you should always remember for 1 glucose, never remember for 1 acetyl CoA. Okay, don't get confused. Okay, now I am asking a simple question. what is the total number of NADH that are produced per oxidation of one glucose molecule by excluding the step of link reaction? What is the total number of NADH that are produced per one glucose molecule by excluding the link reaction?
8.
Correct or not? For one glucose molecule glycolysis I will get 2. Link reaction I will get 2. Krebs cycle I will get 6. I told you to exclude this, then it will be only total 8. Correct na? No point of getting confused over there. So now total how many ATP we produce at gross? 38. But net what I told you? 36 molecules of ATP. Now all of you remember my statements very correctly without having a confusion. I think you all four remember how many number of substrate level phosphorylations occurred? How many? 2. Where? In the glycolysis we produced ATP directly. Which step? Substrate level phosphorylation is a step where there is a production of ATP directly, which means in the 7th step ADP combined with PI, then there is a production of ATP. So here direct production of ATP occurred. Correct na? Here also occurred. So in the entire glycolysis the total number of substrate phosphorylations are how many? 2. Only 2. Correct na? Now tell me in the Krebs cycle for one glucose. One glucose means how many acetyl-CoA? 2 acetyl-CoA. How many ATPs we produced? 2. So total number of substrate phosphorylations for one glucose.
4.
How many? 4. Correct na? You are getting the point. That is also a direct question. Okay. Okay. Slowly think and answer the questions. All of you slowly. Correct. What is the answer for this?
0.
Aram se. The number of substrate level phosphorylation in one turn of
Citric acid cycle.
for citric acid we produce 1 GTP, right? For 1 acetyl-CoA. So 1 GTP giving rise to? That is for how many acetyl-CoA? 1 acetyl. That's why they used the word 1 turn, which means how many? 1. Correct? No, don't be hurry. If I say glycolysis, obviously 2, 7 and 10. Correct na? In the Krebs cycle there is only 1 for 1 turn. Correct na? 1 turn or 1 acetyl-CoA. Then if I ask you for 1 glucose, 4 for 1 glucose. When I use the word for 1 glucose, always remember I am talking about oxidation of 1 complete glucose. When I want to break down 1 glucose, I have to use always 4 steps. Then I have to consider 4 steps. In the glycolysis I have 2 substrate phosphorylation. In the Krebs cycle I have 2 substrate phosphorylation. The total number will become 4. Don't confuse. Now answer this. What is the role of NAD plus in cellular respiration?
Electron carrier.
Now we have to eliminate. It is a nucleo. Is it a nucleotide?
No.
Nucleotide made up of sugar plus phosphate plus base. Correct na? It cannot be the nucleotide.
Not an enzyme.
It is not an enzyme. What it is then?
Electron carrier.
It is a cofactor. What is that? What is called cofactor? Non-protein part. We called as a cofactor. Okay. It is the final electron acceptor. No. So answer is what? It function as a electron carrier. No confusions at all. They are asking which is wrong. So fast. Okay. Even though you know the answer, try to read at least. There is one point in the cycle where FAD plus reduce it to FAD. What is the name of the enzyme that catalyzes this reaction? What is the name of the enzyme? Succinate dehydrogenase. Anyway, this statement is a true statement. During the conversion of succinyl CoA to succinic acid, a molecule of GTP is synthesized. Sometimes they will add the statement by the cleavage of a thioester bond. That is also a right statement. The cycle starts with condensation of acetyl CoA with so it is combined with oxaloacetic acid. Pyruvic acid is how many carbon? 3 plus 2, 5 carbon we can't form because our intermediate should be 6 carbon containing compound. So there are 3 points in the cycle where NAD plus reduce it to NADH2. Correct or not?
Yes, sir.
There are 3 reduction reaction. But if I consider FADH2, there are 4 reduction reaction. Very good. Common. Aram se look into that. Why not pyruvate answer? What is the question? How many times I told you? Just now I told you which of the following biomolecule is common. In the last class, first slide we discussed this only. Common to the respiration mediate breakdown of fats, carbohydrate and protein. Whenever protein breakdown, it undergoes what process? Yes, yes, I am able to see your answers. You can continue. Yeah. What is that? And protein breakdown into amino acids undergo the transamination, giving rise to what? Pyruvate. But pyruvate never enter into the Krebs cycle. It has to convert into acetyl CoA. What happened to the carbohydrates? Giving rise to the glucose, glucose converted into pyruvate, pyruvate converted into acetyl CoA. Correct na? Same goes with fats. Fats undergoes beta oxidation, giving rise to pyruvate, pyruvate gives rise to acetyl CoA. Correct na? So whether it is a carbohydrate, protein or a lipid, ultimately to produce bulk amount of the energy, they have to give rise to acetyl CoA, because acetyl CoA only that enters into the Krebs cycle. Answer is acetyl CoA. No point of confusing over that. We read na?
Lactate fermentation.
Which one of the following process carbon dioxide is not real? See, I'll tell you, everyone is intelligent, but everyone should have a more patience. See, easy questions you can answer easily. When you are solving a tough questions, you will read with the same anxiety. Undoubtedly, you choose it wrong. So, even though you know answer, try to spend a good amount of time on the given question. Okay. Which one of the following process carbon dioxide is not released? So, first pyruvate converted into acetyl CoA in the presence of oxygen. What's happening that time? Aerobic respiration, which means how many carbon dioxide are released? 2 carbon dioxide released. So there is a release of carbon dioxide. Then pyruvate converted into the ethyl alcohol. Correct na? In the absence of the oxygen, again there is a release of 2 carbon dioxide. But when pyruvate converted into lactic acid, this is 3 carbon and this is 3 carbon, there is a no releasing of the other carbon, no carbon is left out. So answer is lactate fermentation. Easy to understand, I think. Slowly. What is your name? Sanskari. Okay. 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 Guess it. Guess it. Yes, 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 yes. Huh? No, why photorespiration will come? We are reading the cellular respiration. What is the question says that? The energy releasing metabolic process in which substrate is oxidized. Oxidized means what? Okay, Hemant. Oxidized because of what? See, let's consider what is fermentation. Pyruvate reduced, correct na, to ethyl alcohol. There is a release of carbon dioxide, correct na. Now the question is here, the energy releasing metabolic process in which substrate is oxidized, means here pyruvate got, sorry, this is oxidation, pyruvate got break down into ethyl alcohol without accepting any electron, correct na. Pyruvate on its own, it is breaking down, and this is only happens in the anaerobic condition. Now you will get a doubt, sir, why it cannot be the glycolysis? Because glycolysis is always happens what? In the absence of oxygen only, and moreover in case of a glycolysis is what happening? Glucose is breaking down into 2 pyruvate molecule. Then what is the exact difference between the, it's okay, what is that simple difference between the fermentation and glycolysis? Tell me, what does, I mean to say, what is the major difference? Fermentation is also occurring in the anaerobic condition, glycolysis is also happening in the anaerobic condition. What is the difference? Because here carbohydrate is breaking down into 2 pyruvate. Here the end product of the carbohydrate metabolism only breaking down into alcohol. You are getting? And why we used the word energy releasing metabolic process? See, wherever you see the fermentation process, it is always a energy releasing metabolic process, and this is the only process where there is a no external electron acceptor that accept the electrons. In simple words, wherever you heard the energy releasing metabolic process, always remember it is the fermentation. In the upcoming slide you will understand. This we will discuss it, but anyway you can answer it. Hey, what happened to this? Anyway, what is the question? Aerobic respiratory pathway is approximately termed as amphibolic. Why we use the word amphibolic? Amphibolic is a pathway of what is called catabolism? Breaking down. Anabolism, what we discussed? Building up. Correct na? So, in the catabolism, if you consider the Krebs cycle, there are many products break down and there is a production of many other products. And very important statement, most of the people will miss it. The Krebs cycle, it is called it as the amphibolic pathway. One of the major reason, whatever the end products, whatever the intermediate products that produced in the Krebs cycle required for the biosynthesis of other. Let's take an example. We produced what? Citric acid. And what else we produced? Alpha ketoglutarate acid. What else we produced? Succinic acid and malate. Fumarate. Actually this succinic acid, malic acid, they are also required for the metabolism of nucleic acid. They are also required for the metabolism of proteins. They are also required for the metabolism of amino acids, which means whatever the intermediate products that are produced in the Krebs cycle used for the biosynthesis of other products where both catabolism and anabolism is happening. That is the reason we call it as the amphibolic pathway. So 90% of the students remember all this, but it is more important we have to remember the structures and most importantly enzymes. Okay, correct. Now look into the point without having confusion. Everyone read those steps. I encourage everyone to remember the enzymes, compulsory for the exam. Okay, just give me a minute. Anywhere here, nucleic acid, it is Krebs cycle. Anyway, not a problem, we can continue here. Now first, what is happening? Oxaloacetic acid combines with the acetyl-CoA, which is a 2-carbon containing compound, and this reaction can be catalyzed by a versatile enzyme, citrate synthase enzyme. So friends, tell me what is happening here. We are just adding the acetyl group to the oxaloacetic acid. So it is an acid, exactly to that acid functional group only we add this particular acetyl, and here there is a addition of the water also. So whenever we are adding the water, is it a condensation or decondensation? What is called condensation first of all? Condensation means loss of water, correct na? When you are adding the water, you should not use the word decondensation, use the word hydration. Okay, there is a difference. Condensation means, in a simple words, remember, releasing of water, H2O, a simple word. But when I am adding the water, we call it as the hydration, correct na? We don't use the word we are removing the water, we say that there is a release of water. Anyway, we will quickly summarize this. Then the next second enzyme, citrate is converted to isocitrate. So what is happening? Both are the isomers. The enzyme involved is cis-econitate enzyme. Again, for the exam point of view, what you get? What is the major cofactor, which is the Fe2 plus. Now isocitrate is how many carbon? 6 carbon. It is undergoing dehydrogenation or oxidation to give rise to oxalosuccinate. That is also a kind of oxaloacetate is how many carbon? That is also 6 carbon only. Later it is hydrolyzed back, break down to alpha ketoglutarate, which is how many carbon? 5 carbon. Anyway, remember, whenever isocitrate is converted into the alpha ketoglutarate is a 5 carbon containing compound. So we said it to be the first decarboxylation reaction and first reduction reaction, where there is a production of reducing equivalent, that is NADH or NADH2 you can consider. Next, alpha ketoglutarate converted to succinyl-CoA. Again, alpha ketoglutarate is a 5 carbon, succinyl-CoA is a 4 carbon. Again, there is a loss of 1 carbon. Undoubtedly, we call it as the decarboxylation. Again, there is a second reduction reaction. There is a production of NADH2. Now, later what is happening? Now, remember for the exam. What is the first enzyme? This one. Isocitrate dehydrogenase converted to the alpha ketoglutarate acid, which is the first reduction reaction. Now, please focus everyone. The second reduction reaction, that is what? Alpha ketoglutarate converted to the succinyl-CoA. The enzyme again, alpha ketoglutarate dehydrogenase enzyme. Now again, question for the exam. TPP, lipoic acid and magnesium. Anyone tell me where we used these two? Very, very good. See, this is the exact question which is asked in the AIMS exam previously. Which of the following two enzymes having the same cofactors? Now look at this. Pyruvate converted into acetyl-CoA by an enzyme called as pyruvate dehydrogenase enzyme. Correct na? It is having E1, E2, E3. Similarly here, alpha ketoglutarate converted into succinyl-CoA. The enzyme involved here, alpha ketoglutarate dehydrogenase, that is also having E1, E2, E3. But the names are little different, but they are dehydrogenases and transacetylases only. But they are also using the same cofactor. This only you have to remember. Everyone can easily remember the calculations. Then succinyl-CoA is how many carbon?
5 carbon.
Succinyl-CoA is a 4 carbon converted into succinate. What is breaking? CoA is breaking. CoA contain what? Thioester bond. Because of that thioester bond only, there is a production of what? GTP. Now what happened to this GTP? GTP transferred its phosphate group to ADP. Then upon transferring the phosphate group, GTP come back to GDP. Correct na? Then ADP converted into ATP. Correct na? Why? All the cells mostly uses ATP as the direct energy source because they are having the binding sites majorly for ATP. Don't get confused. Then what is happening? Succinate converted into what? Fumarate. Correct na? Succinate is how many carbon? 4 carbon. Correct na? Again, succinate converted into fumarate by dehydrogenation. When I use the word dehydrogenation, enzyme is dehydrogenase enzyme. But here reducing equivalent is FAD plus converted to the FADH2. Then fumarate, which is the 4 carbon converted into malate. Both are 4 carbons only. So here fumarate added by the water. Hydration we can say. Then it is converted into the malate. Now malate is 4 carbon converted into oxaloacetic acid. Again by an enzyme, malate dehydrogenase enzyme, where there is a production of another NADH. Now, if you miss the questions from here, again there is a great possibility that question can comes from the enzymes and regulation. How many regulatory enzymes are there? In glycolysis? 3. Number one, first step, third step, 10th step. Tell me what are the enzymes of Krebs cycle?
Hexokinase.
Krebs cycle. Krebs cycle.
First step.
What is the first enzyme? First, what is the first step?
Acetyl.
First step, acetyl CoA combines with oxaloacetic acid. There is a production of citric acid. Correct na? What is the name of the enzyme that is involved in the synthesis of, Aram say, involved in the synthesis of citric acid? Very good. What is that? Citrate synthase. This is one of the regulatory enzyme. Correct na? Then isocitrate converted into isocitrate. Later isocitrate converted into ketoglutarate. What is the enzyme? Isocitrate. Always remember the short forms. IDH. Isocitrate dehydrogenase. This is the second regulatory enzyme. Number 1, citrate synthase. Number 2, isocitrate dehydrogenase. Now alpha ketoglutarate is converted into succinyl CoA. What is the name of the enzyme?
Alpha keto.
Glutarate dehydrogenase. Same. So simply you can remember alpha ketoglutarate dehydrogenase, KDH. This is third regulatory enzyme. Correct na? These 3 are said to be the major regulatory enzymes of the Krebs cycle. Every enzyme is important, but these 3 are regulatory enzymes because they are going to perform irreversible reactions. Correct na? Now, tomorrow we will discuss the electron transport chain, which is the most important one. Now all of you quickly answer, in case of a glycolysis we produced NADH2, and in the Krebs cycle we also produced FADH2 and NADH2. But so far when we consider glycolysis, link reaction and Krebs cycle, hardly you can see 2 ATP directly. Correct na? Remaining ATP you can't see directly. Remaining ATP is in which form? NADH and FADH2 form. Just now we discussed that they are acting like what? Of course GTP only. Okay. Let's consider, no, I am asking direct ATP, I am not asking conversions. Okay. When we say conversion, then we can consider 4. Okay. And that's what I use it now only in the glycolysis. You can use 2 ATP. That is also true. We can say 4 ATP when there is a conversion. Or we can say generally net visible is only 2 of glycolysis. Again, if they use the word net visible of ATP for 1 glucose, then we have to consider GTP also. Then we will say 4. I believe everyone is understanding correctly. Net visible ATP for 1 glucose is 4. Net visible ATP in the glycolysis is 2. Correct na? But we are getting NADH2, we are getting FADH2. Most of the energy is present here. Now we already calculated na, per 1 glucose how many NADH produced? Per 1 glucose. Don't confuse. I am using the word 1 glucose, which means Glycolysis, we have to consider link reaction, Krebs cycle. Then how come 8? Then 10, na? 10 in DH. How many FADH2?
2.
Correct, na? So, almost 30 ATP is present here only. Here 4 ATP is present, which you can't see. Correct, na? Now, what we have to do it, we have to reduce them, we have to oxidize them and we have to produce ATP. You are getting? So, if I want to break down them, first there should be a requirement of someone who accept it, because they are breaking down and releasing electron, and that electron someone has to accept. If someone wants to accept the electrons from them, first these has to travel from where to where? Where the Krebs cycle occur? Krebs cycle happened in the mitochondrial matrix. We'll see in the next class. From the mitochondrial matrix, they have to move to where? They have to move to where? It's over. Okay. They have to move to where? They have to move to electron transport chain. Where the electron transport chain is present? In the inner membrane of mitochondria. So, which are moving? NADH2 and FADH2 are moving. They are acting like what carriers? Electron carrier. That's why name of the chain is electron transport chain. No point of getting confused over there. Okay. So, this is about Krebs cycle as well as the and glycolysis. In the next class, we will complete the electron transport chain very elaborately. You are from which college?
Me from Pune.
Oh, great. You came today itself. Okay. Okay. Revise everyone this completely, and in the tomorrow class I will complete the electron transport chain under oxidative phosphorylation. 90% the tough questions comes either from Krebs cycle or from the electron transport chain. Correct, na? Don't get confused. After completing of this, we'll start the plant growth and development. Consider by Saturday it can be completed. Then we'll solve at least 250 to 300 MCQs exclusively from these 3 topics. By the plant physiology will be completed. Okay, done. Okay, revise this. Thank you. All right, thank you. All right, okay, come. You come, man, I'll give you the access. Come dear, you also.